All right, you are in the right place, and today we are going to talk about CyCombine for Dummies. And the reason we're talking about this is it's an overview of how batch effects in cytometry can hurt reproducibility and how normalization can help. And just as a reminder, our mission at TACO is to make high parameter cytometry routine and simple for clinical trials. So who might care about this problem? Drug developers who are familiar with the idea of batch normalization and who want to know what the algorithms are doing with the data. They think of these algorithms as black boxes and want to know underneath the hood what's going on. And just as a reminder, we have a batch norm video also on YouTube, but I'm going to give you a quick refresher as to the underlying purpose. Short version, the goal of batch normalization is to reveal true biological signals and get rid of potentially false positives and false negatives. So the status quo with no batch normalization is you spend millions of bucks on a cytometry data set. You could potentially pursue a false signal due to technical variation. Before batch normalization, you've got a bunch of different cell types here on the x-axis. On the y-axis, you've got the p-value. 0.05 is a statistical significance threshold. Now, after normalization, all of these cell types' p-values have shifted, and batch normalization could potentially have had a false positive here, which is now removed, or potentially revealed a truly significant result. So this allows you to get true insights on your data set and chase the ones that really matter as opposed to potentially a wild goose chase. So if it's so good, why isn't batch normalization used on trial today? And this is what was asked in CYTO2024 in a great technical series called Potential and Challenges of Clinical High-Dimensional Flow Cytometry. And number 1 and 2 were, I don't know how to assess the output of these algorithms and I don't like the idea of manipulating data. So we're going to try to go and peel back the layers behind the black box and show you a little bit about what's happening to the data underneath the hood. So how does it work? Today we're going to talk about CyCombine, which is an amazing paper that came out in 2022, and this has 3 major steps: normalizing the data, using clustering to group similar marker expression, and then using a technique called combat to remove batch effects. So again, there are 3 steps here. Normalize, we're going to standardize each batch data using a z-score, which is just simply subtracting the mean off and dividing the standard deviation. Clustering, this is a pre-processing step using what's called a self-organizing map to cluster cells into groups, and this helps capture biological diversity. This allows you to potentially get T cells together and memory-naive cells together, so you capture that biological diversity. And then correction, this is the true engine where within each cluster, you can remove batch effects and maintain these biological signals. So this is an example of a marker before batch normalization. You've got CD11b expression over here. From the X to Y axis, it's basically a density plot. This is the exact same sample ran in 4 batches, 1, 2, 3, and 4. And this is a batch effect in that you would expect all of these guys to be on the exact same curve. And the goal of batch normalization is to get them all on that curve. So we're going to kind of go through this, all these steps: normalization, clustering, and correcting, so you can see exactly what is being done to your data. First, let's go to the dummy dataset assumptions. You're going to have the same 3 specimens run 4 times. So for example, the dummy dataset, you have 15 cells per specimen, obviously in flow cytometry experiments in the hundreds or potentially millions of cells. 3 specimens per batch, 4 batches, 5 markers per panel for a total of 180 cells that are in this experiment. And then within the markers per panel, we just made up a dummy dataset, but feel free to use your own from CD45 to CD28, so it's grounded in some biological example. So remember, 3 steps: normalize, cluster, and correct. So let's go through the normalization step. So first, you begin with your raw data. So you list out all of these 180 cells: batch 1, specimen, CD45, all the way to CD28. And here you can see all the raw intensities, and this is the beginning. And now we normalize each marker expression value. So we calculate what's called a z-score for each raw marker expression value. So this is calculated on a per marker per batch basis. You take the raw intensity, you divide by the mean intensity, you take the raw intensity, subtract the mean intensity, and divide the standard deviation. Let's see this in action. You can calculate the z-score for each raw marker expression value. So for example, you've got cell 0 through cell 179 over here, batch 1, specimen 1. Applying this formula to each of these markers, you get a normalized value. So this is the first step of pre-processing. The next step is clustering. So normalizing, clustering, and correction. We're on the clustering step now. So now that the cells are normalized, you can sort them into clusters on a grid known as a self-organizing map. And basically, each cell is going to be put in one of 64 buckets. And we're going to walk through what this technique is. It's called SOM. And without this kind of technique, batch correction might treat all cells as a single group, and this could ignore differences between cell types like naive or versus memory T cells. So what we do here is put each of these cells into a specific cluster and then do downstream analysis on it. So how does that work underneath the hood? Basically, we create this 8 by 8 grid, so starting from 0, 0 over here, going to 7, 7 here, so this is 64. And then we initialize every single neuron here with a 5-list vector of random values. And we're going to do that for all 64 of these neurons. And don't worry too much about how these are initialized, but just know that this grid is initialized with a bunch of random values. And so, for example, neuron 0 might be 0.1, negative 0.5, 1, negative 0.8, 0.3. This neuron over here, 3, 4, might be 0.2 to 0.3. And so during the training, these values are going to be updated to better align with the data. We're not going to go through that in exquisite detail here, but we're just going to give you a sense of what's happening. So the first thing that happens is what's called a best matching unit step. So if you remember, we had cell 0 through cell 179. We're going to take cell 0. Remember, we've got all the markers here and all the expressions here. And we're going to compare cell 0 to every single neuron across this entire 64 unit. For example, we have neuron 16. You see a computed value D equals 1.2. And a carefully attentive reader might say, what is this D number? This D stands for distance. This is the Euclidean distance, computing the difference between this vector, 0.9, all the way to this vector. We're not going to go to that equation in detail, but suffice to say, this is a distance function. And then we're going to go through that for another neuron, and you can see D equals 1.5, 2.0, 3.5, etc., through all 64 of these neurons. So ultimately, for cell number 0, what we're trying to find is the lowest distance, so the best matching unit. And so for all those neurons, 0, 0, all the way through 64, we're just showing a subset here. Each one of them has a distance value, 3.59, 2.03, 1.16. And what we do is we find the best matching unit. So the neuron with the smallest distance becomes the best matching unit for the cell. That cell is mapped to the corresponding grid location of the best matching unit, and then we rinse and repeat through all 180 cells, and we keep updating and aligning and making sure that this gets accurately captured. So the end result of this is that the cells are clustered and assigned. So for example, cell 0 is assigned to cluster 50, cell 178 is assigned to cluster 21. So now you have a bunch of different cells in different clusters. And now we're ready for the correct step. So we did 2 pre-processing steps. We normalized the data, we clustered the data, and now we're ready for batch correction. And so prepare for combat. This is a 2007 paper which really pioneered one of the fields of batch correction, and we're going to walk through some of the math here in a little bit of detail. The premise of combat is that batch effects occur in 2 cases. One is there's an additive batch effect, meaning things are systematically shifted to the left or right. Or 2, there's a multiplicative batch effect, which is either the range is too compressed or too spread out. And you can see the equations over here. We're not going to go through the underlying total equations in great detail, but we're going to give you a sense of the intuition behind these. So let's take the additive effect. So for example, for marker 1, if batch 1 measures slightly higher values, for example, an average 0.5 higher than the overall data set, we subtract 0.5 from all the values in batch 1. Similarly, if batch 2 measures slightly lower values, for example, it's on generally less by 1.0 compared to the overall data set, we add 1.0 to compensate for it. So this additive seems pretty easy to understand, which is it's off to the left or right a little bit, so we adjust. The multiplicative effect is a little bit more complicated. Here's the dictionary definition over here, but let's just go through the intuition. If you're looking at a marker in 3 batches, batch 1, marker 1 has a small range, so it's compressed, 10.5 to 11. Batch 2, that very same marker has a wide range, 2 to 30. And batch 3, marker 1 has a moderate range, 7 to 13. So intuitively, we want to spread out this, compress this, and maybe tighten this up a little. And so that's exactly what we're saying here, which is without correction, if you don't compare, it could be potentially misleading. So this multiplicative effect scales the variability. Batch 1 is stretched out to match the global variability, batch 2 is compressed, and batch 3 is adjusted slightly if needed. So now let's zoom in and apply this to cluster 50. So we go over here, we've got a 64 grid, we have dozens of different cells over here, and we're going to pick a specific marker over here, like 1,6, which is cluster 50. And remember, within this cluster, there are going to be a bunch of individual cells. So within cluster 50, you have cell 0, 1, 2, and you have a bunch of different readouts and normalized expression values here. And now what we're going to try to do is get to a corrected value, which is ultimately what this entire exercise is for. We have this normalized value, we're going to have this additive batch effect, a multiplicative batch effect, and then arrive at a corrected value. And to do that, there's a big equation. This is a very fancy equation, and we're going to break this down step by step. The left-hand side of this is the ultimate goal. This is the corrected value for this particular cell. And the first step over here is a standardized marker value. So that normalized thing, we subtract the mean, we divide by the standard deviation. Then we subtract off the additive batch effect, which is the mean offset for that specific batch. Then we're multiplying this parentheses over here by the global standard deviation of the marker G before batch correction, and then we're dividing by the square root of the multiplicative scaling factor for this batch. And finally, we're adding back the global mean of the marker G. So then we get a corrected value that we can read for this particular cell. So let's see that in action. For this cell, for this cluster, we have a bunch of different expression values. We apply this additive batch effect within the cluster, the multiplicative batch effect within this cluster, and we end up at a corrected value for each of these cells. So for example, 2.50, 2.107, etc. So now you've got a bunch of corrected values. So what did we just do? We went through normalization, clustering, and finally this correction step. All of this is in service of ultimately getting really high quality data. This entire exercise is to avoid pursuing millions of dollars or spending millions of dollars pursuing a fake signal or potentially missing a real signal. If you don't use batch normalization, you could potentially see a curve that looks like this. And the goal of batch normalization is to get rid of those false positive signals and potentially remove technical noise and reveal true significant values in your data set. So if you're interested in other details like performance measures, how exactly do we know that batch normalization is working, subscribe to our YouTube channel to learn more, and let us know in the comments or DM if there are any topics you want us to cover. Thanks for stopping by.